Continuing on chapter 16, we're on part 4, cell signaling. So we're going to be continuing looking at specific aspects of two pathways, the G protein coupled receptors and enzyme coupled receptors. In the last video, part 3, I described a G protein coupled receptor pathway resulting in glycogen breakdown. Here, I will demonstrate how we can alter this pathway to get a different cellular response. We're going to see that the signaling components downstream can vary from cell type to cell type, so even if you're using the same ligand and receptor, you may have a different response in the cell depending upon the components that are activated. So here's the pathway that I finished up with in the last video where we're looking at a cell, got the cell membrane, got my G protein coupled receptor with its 7 passes of alpha helices bound to its ligand, which is in this case adrenaline, activating the alpha subunit of a trimeric G protein. When it's active, GTP enters the binding site, and the alpha subunit activates an enzyme called adenylyl cyclase, which makes a second messenger called cyclic AMP, and cyclic AMP binds to and activates a kinase called protein kinase A. And through a few more steps, activates the process of glycogen breakdown, which would release glucose for the body. We're going to see that this pathway can also diverge and at pKa specifically. So we would begin with the same ligand, receptor, alpha subunit, activate adenylyl cyclase, make cyclic AMP, activate pKa. But in some cells, rather than phosphorylating phosphorylase kinase, the target of pKa can be in the nucleus. So here, this is supposed to be the nuclear envelope, and pKa is entering the nucleus, and now it has a new target protein for phosphorylation, something called CREB. CREB is a transcriptional regulator. When pKa phosphorylates CREB, CREB will bind to the regulatory region of very specific genes, so some genes are regulated by CREB, and then when we see the arrow here, this suggests that the genes are now being transcribed. So transcription of target genes. This is not a general transcriptional regulator that is used for every single gene. This is a gene-specific transcriptional regulator. There are going to be genes that have specific regulatory sequences to which the protein CREB can bind, and only those will be regulated by CREB. This is going to be a slow response. So we had divided the responses into short-term, kind of quick responses, fast and slow responses. Glycogen breakdown, where we're just activating an enzyme, that would be one of those shorter responses, whereas the response dependent upon CREB is going to mean that before we get a new protein being made, we have to transcribe that protein, translate it, and then properly fold it and activate it. So this is going to be a longer-term process on the order of minutes to hours. And here's the image from your textbook showing you the same thing. I've just given you the name of a particular transcriptional regulator, CREB, but it's a similar thing to what you're seeing in your textbook. So here we have the same pathway to show you. They're not giving you what's upstream, but you can see where they're starting with cyclic AMP, activation of pKa, translocation into the nucleus, phosphorylation of its target, and transcription of target genes. This isn't the only place where the pathway can diverge. So I've got, going back to my image where I'm trying to put them all in one place, we had our original adrenaline binding to our G protein coupled receptor, activating the alpha subunit, and this was, at this point, activating adenylyl cyclase, making cyclic AMP, which activated pKa, which could do one of two things, either stimulate glycogen breakdown and or transcription of target genes through CREB. However, this pathway can diverge earlier in some cell types, and rather than the activated alpha subunit targeting adenylyl cyclase, its target is another enzyme called phospholipase C. What phospholipase C does is target an inositol phospholipid. This is a molecule that is made of a lipid, this diacylglycerol. It's what keeps it in the cell membrane, and a sugar. This is the inositol. So it's a lipid stuck on a sugar. Phospholipase C is an enzyme that's just going to break the connection between those two molecules. The sugar, diacylglycerol, which is abbreviated as DAG, it has this kind of tuning fork type of shape, and then we have the sugar. So the IP3 will separate from the diacylglycerol, and because it's no longer stuck to a lipid, it can move into the cytoplasm. When IP3, which is what it's now called, when IP3 moves into the cytoplasm, it can interact, I'm now showing you the IP3 is this little spot here, with an ion channel. So the IP3 becomes a ligand for a ligand-gated ion channel. And actually what's not written here, and maybe I'll add in, that this is the ER, is what this is supposed to be. So this is the endoplasmic reticulum. It's shown kind of small, but just so I can fit it in there. And the endoplasmic reticulum has a calcium channel. The calcium channel is opened by IP3. It cannot open the calcium channel while it's stuck to diacylglycerol, so it needs phospholipase C to release it. The IP3 binds to the channel, opens it, and because calcium is highly concentrated in the ER, it is released into the cytoplasm. It diffuses down its gradient. And then together, the calcium and the diacylglycerol activate another kinase called protein kinase C. So this is a third pathway and another kinase that is activated. And this is the image from your textbook showing the same thing, activating the alpha subunit, which turns on phospholipase C. Phospholipase C breaks the connection between diacylglycerol and IP3. IP3 is no longer tethered to the membrane. It can activate the channel, and together they can activate PKC. I also have a video showing this process, and I'm going to run that in a moment.
Phosphatidylinositol 4,5-bisphosphate, or PIP2, lies at the heart of many signaling pathways, and it is typically activated by upstream signaling through G-protein-coupled receptors, or GPCRs. The process begins with the activation of a GPCR by a signaling molecule. The GPCR undergoes a conformational change and subsequently activates a trimeric G protein, GQ. The alpha and beta-gamma subunits of the GQ protein dissociate and work together to activate phospholipase C beta. Activated phospholipase C beta now splits the PIP2 molecule into two fragments, soluble IP3 and diacylglycerol. Both of these fragments relay the signal onwards. IP3 diffuses through the cytosol and binds to calcium channels in the endoplasmic reticulum membrane. When IP3 binds, calcium channels open, releasing calcium ions into the cytosol. This rise in the cytosolic calcium concentrations causes protein kinase C to move to the plasma membrane, where it is activated in turn. Protein kinase C activation requires both binding to diacylglycerol and the continued presence of calcium ions. Once activated, protein kinase C phosphorylates target proteins that vary depending on the cell type.
We can see changes that occur during cell signaling if we tag proteins in live cells with fluorescent proteins that we can see. So in this case, in these experiments described from a publication in 1998, we see live mouse cells that are stuck to a slide. And they do have PKC in them, but you can't normally see protein kinase C. So what they've done is added a sequence for PKC, so DNA with a sequence for PKC that kind of runs right into the sequence for green fluorescent protein, or GFP. This would be called a fusion protein. So when this is transcribed and translated, the instructions for the two proteins are together, and as PKC is translated, GFP is also translated. So wherever PKC is, so is the green fluorescent protein. So if this image right here, D, is of a bright field image using a typical light bulb, in the middle panel we would be using a fluorescence light source. And we can see that two of the cells have taken up the construct and are glowing green, And they're glowing green throughout the cytoplasm. We can see a little bit of a less dark space where the nucleus is. If we add a ligand for the receptor and stimulate the pathway, we'll see we get a translocation or a movement of PKC to the cell membrane. And now the cells have kind of like a highlighter mark where the PKC has moved. When I was a grad student, these are the type of experiments that I like to see because it wasn't all happening in tubes of clear liquid. We could actually see the cells and watch changes happen within the cells. So I wanted to put a little bit of context into how cells can use calcium as an intracellular signaling. And it's nice that the textbook uses an example from developmental biology. It's very much up my alley. We can see that a wave of calcium is released upon fertilization. The specific example they're going to give you is a sea urchin egg, but we also have a wave of calcium release in human fertilization. We are going to see that the sperm binding to the egg, the sperm acts as a ligand for a receptor on the egg that initiates calcium signaling. And what the calcium signaling will do in humans is going to cause a completion of meiosis. Our eggs are actually paused in metaphase 2 of meiosis until fertilization. Well, then, once fertilization has occurred, the embryo needs to start cell division, and a dividing embryo is going to need quite a lot of transcription and translation to produce new proteins. And there's something also called polyspermy that should be blocked, and we're going to look at that in the specific case of sea urchins. Polyspermy is more than one sperm fertilizing the same egg. So the sperm you're going to think of as the ligand, and it binds to a receptor on the egg that activates phospholipase C. And we get a release of IP3 and diacylglycerol, which results in calcium being released from the ER. In the picture below, they're showing an egg with a dye that shows you the intensity of calcium signaling. So we have the image here, but I'm going to show you a video of that with more detail.
When a sperm cell fuses with a sea urchin egg cell, calcium ions begin rushing into the cell at the site of fusion. In these experiments, calcium concentrations are visualized and measured with a fluorescent dye that becomes increasingly brighter the more calcium is present. Brightness is then translated into a color scale, and, in this three-dimensional display, into peak heights, where red and high peaks represent the highest calcium concentrations.
What does the calcium wave do? Turns out that underneath the cell membrane, eggs have vesicles waiting right underneath that cell membrane, kind of the way there are synaptic vesicles waiting at the end of an axon. Stimulation of the egg by the sperm and release of calcium causes exocytosis of these vesicles. And we can actually see the consequences of this exocytosis in sea urchins, and I'll show you a video of that in a moment. And we get a lifting of something called a fertilization envelope that stops any more sperm from coming in and fertilizing the egg. You don't want to have polyspermy, right? Having an extra of every single chromosome would be fatal. So in this movie, we'll see each egg being fertilized separately. First the one on the left, and then the one on the right. And as they're fertilized, you'll see a fertilization envelope developing. I'll play them once at the speed that's given, and then once I'll slow it down. So now I'm going to play this at half speed. Then I'm going to show you a similar video, but looking at fertilization and the calcium wave at the same time. We'll see in this very quick recording, they're going to show you fertilization happening on the left side, where you can see the lifting of the fertilization envelope, and on the right, the wave of calcium coming through the egg.